Under a microscope, a glowing thread spins slowly in a circle. It's attached to a single molecule, taken from inside cells like yours. And that molecule is a motor. I want to be precise about the word motor because biology uses it loosely, and this isn't one of those cases. This is not a machine in the sense of a complicated system. It's a machine in the sense of a rotating shaft turning inside a fixed housing, driven by a flow, doing mechanical work. When researchers described it in Nature, they called it a rotary motor, the smallest known. There are thousands of them in every mitochondrion in your body, and they are running right now. Let me build up how it works because the whole design is remarkable. Start with what I talk about constantly on this channel. Your electron transport chain takes electrons from your food, passes them down a series of complexes, and uses the energy released to pump protons across the inner mitochondrial membrane. That pumping creates a difference. More protons on one side than the other. And since protons carry positive charge, that difference is both a concentration gradient and an electrical voltage. Together they're called the proton motive force. It's stored energy, like water held behind a dam. Now that membrane is otherwise sealed to protons. They can't simply leak back through. There's essentially one door. That door is this motor. Protons flow through a channel in the part of the machine embedded in the membrane. And as they pass through, they force a ring of subunits to rotate. Attached to that ring is a central shaft. It's about 1 nanometer in radius, and it turns inside a barrel formed by 6 other subunits about 5 nanometers in radius. Let me put those numbers in perspective. 1 nanometer is a millionth of a millimeter. This is a drive shaft roughly the width of a few atoms. Now, what does the rotation actually do? The barrel around the shaft contains 3 catalytic sites. The shaft isn't smooth. It's asymmetric, so as it turns, it pushes against each of those 3 sites in sequence, forcing each one to change shape. And those shape changes are what produce ATP. Each site grabs ADP and a phosphate, and the mechanical squeeze from the passing shaft clamps them together into ATP. 3 sites, 1 full rotation, 3 ATP molecules. So the whole thing is a water wheel. Flow drives rotation, rotation drives chemistry. Your body converts a proton flow into mechanical rotation and then back into chemical energy, and does it billions of times a second across your cells. Now, that was the theory for a long time before anybody had seen it. The rotation was proposed on the basis of crystal structures and biochemistry. It fit the evidence beautifully. But nobody had ever watched a single molecule turn, because molecules are far too small to see rotating under an optical microscope. In 1997, a team solved that with an approach I find genuinely delightful. They took the motor and fixed it upside down onto a glass surface, anchoring the barrel so it couldn't move. Then they attached a fluorescent filament to the shaft, a long glowing marker sticking out sideways big enough to see. Then they added fuel and watched. Spun. The filament rotated for more than 100 revolutions, always in the same direction, exactly the direction the crystal structure predicted. That's the footage you'll find if you go looking. A little glowing line turning steadily, driven by one molecule. And here's the detail I most want you to have because it's the kind of thing that gets lost when a fact travels. In that footage, the rotation looks slow. A few revolutions per second. You can follow it comfortably with your eye. That's not the real speed. The problem is the marker. The filament they attached is enormous compared to the motor. It's like bolting a telegraph pole to a hand drill. Dragging that through water creates so much resistance that the motor is essentially stalled by its own instrument. When researchers calculated the true rate from how fast the enzyme actually works, the estimates came out between 200 and 500 revolutions per second. Other measurements have it saturating at around 130. So the beautiful videos everyone shares are of a motor running with the handbrake on. The real thing turns something like 50 times faster than what you're looking at. I like that for 2 reasons. First, it's a nice reminder that a measurement can change the thing it measures. And second, the honest version is more impressive than the version people repeat, which doesn't happen often in this field. There's one more property worth knowing, because it tells you something about how your cells handle emergencies. The motor is reversible. Run it forwards and proton flow drives rotation, which makes ATP. Run it backwards by feeding it ATP instead, and it hydrolyzes that ATP, rotates the opposite way, and pumps protons out across the membrane. Same machine, either direction depending on which side has the surplus. That's not a design flaw. A cell that's temporarily short of oxygen can burn ATP to hold its membrane voltage up, keeping the system intact until conditions improve. It's expensive, and it's better than the alternative. Alright, what do you do with this? And here I want to be straightforward with you about something because you may have noticed a pattern in these videos. My answer is going to be the same as it always is. Move. I'm not going to dress that up as a new insight, and I'm not going to invent a special protocol for this particular machine because there isn't one. Here's the actual reason, and I think it's more interesting than a novelty tip would be. This motor has one input: proton flow. Nothing else. And proton flow is determined by how quickly protons are being consumed, which is determined by how fast the motor is turning, which is determined by how much ATP is arriving, which is determined by how fast you are spending ATP. That's the whole control system. Demand. You cannot speed this motor up by adding something to it. You cannot instruct it. You can only ask for more output, and the only way to ask is to spend ATP. Muscle contraction is far and away the largest voluntary way to do that. So when I keep landing on the same answer, it's not a shortage of ideas. It's that I keep arriving at a machine with a single control. Now, where my own recommendation sits, and it stays narrow as always. This motor runs on pressure that something else builds. That something else is the electron transport chain. CoQ10 is one of the carriers within it, moving electrons between complexes so protons can be pumped. Levels tend to decline as we age. I am not claiming that any supplement makes this motor turn faster. The speed is set by demand, and I just spent a minute explaining why. Implying otherwise today would be an easy sell, and it would be false. If you want to support the machinery that builds the pressure, the link in the description below is an affiliate link to Mitochondrial, which is PQQ and CoQ10 with a 90-day money-back guarantee. And if you like the version where the famous video comes with an explanation of what it's actually showing, subscribe. This video is educational only, and it's not medical advice. Talk to your doctor before starting anything new.